Hello students, I am Jinesh Udupi. We will continue with the paper Molecular Biology, Evolution and Ethology. This is session 7, and in this session we are going to study the biotechnology topic, and we are going to study about two things, that is transgenic animals and monoclonal antibodies. So biotechnology. Now what is biotechnology? Biotechnology is a field of biology, and it mainly deals with the genetic material and the techniques, etc. So by definition, it is utilization of biological process, organism or system to produce products that are anticipated to improve human life is termed as biotechnology. So what we do in biotechnology is we are going to use the biological process, or we are going to use the organism or a particular system, biochemical system, and our aim is to produce the products which are going to improve the human life. So that is what we do in the biotechnology. And you can also define biotechnology as any technological application that uses biological systems, living organisms or derivatives thereof to make or modify products or processes for specific uses. Now, biotechnology involves so many different kinds of techniques. So here we are just interested in knowing what is biotechnology and what are the uses of biotechnology. So the techniques and other things, it is not in the scope of this particular topic, right? So applications of biotechnology. Biotechnology is used in the development of antibiotics, vaccines, gene therapy, etc. So therapeutics. In the field of medicine, biotechnology is being extensively used to produce monoclonal antibodies, vaccines, gene therapy, etc. And as far as diagnosis is concerned, biotechnology is proving very helpful in developing different techniques which help the doctors to diagnose the diseases accurately and at a short time. As far as industry is concerned, biotechnology is also used in food industry to produce wide range of food products. Then as far as agriculture is concerned, so biotechnology is used in improving fertilizers, beverages, and different ingredients of food products. Now, regarding the environment, biotechnology is used in waste treatment techniques and bioremediation and energy production. So, producing energy from the biological waste, etc. So, there the biotechnology is being applied. So, biotechnology also has its applications in the field of agriculture, forest, and horticulture, right? And for example, all of you heard about the BT cotton, right? So the gene, they have used a gene from Bacillus thuringiensis bacteria, and they have produced a cotton variety which is resistant against cotton bollworm, right? So like this, you can think of the application of biotechnology in improving the quality and improve the resistance of plant against the diseases, etc. I have just given one example of BT cotton. In the same way, using biotechnological tools, they have produced golden rice, where they have inserted the enzyme which is responsible for the production of vitamin A into the rice plant, right? So what is happening is that particular plant will produce the rice seeds, which is very rich in vitamin A. So like this, the biotechnology has many applications in the field of agriculture also. And uh, chemical intermediates. So in the preparation of chemical intermediates also it is used. And since the techniques need some hardwares, so the development of equipments and hardware related to the biotechnology is also can be considered as one of the applications of biotechnology. Transgenic animals. So transgenic animals are the animals which carry foreign DNA in their cells. The foreign DNA or the gene from other organism is inserted using the recombinant DNA technology. So, in the biotechnology, what they do is they will take out a gene from one organism of a particular species because that gene is of some interest, because it has some value, commercial value, economical value, medical value, whatever it is. So they will get the gene from one organism and they will introduce that gene into another organism, right? So the organism which receives the foreign DNA, which has the foreign DNA in its cells, and it is also producing the product of that foreign gene. So such animals are called transgenic animals. So in the previous slide, I have given the example of BT cotton, Bacillus thuringiensis. So what they have done, they have taken a gene from bacteria and they have inserted that gene in the cotton plant. So now the cotton plant is producing a particular product of bacteria. So that is an example of transgenic plant. In the same way, scientists have developed animals which have foreign DNA in their body and they produce the substance which is not native to their body. Okay, so that's how they produce transgenic animals. And remember, the product of transgenic animal normally improves the quality of that particular animal, whatever the economical purpose. So their intention is to improve the quality of the product. So a transgenic animal is one whose genome has been altered by the transfer of a gene or a genes from another species or breed. Okay, that's about the transgenic animals. Many examples of transgenic animals are there, like fishes and goats. What they have done is they have inserted the gene to improve the quality of wool uh, from another organism into the goats. And as far as cattle is concerned, so they have used some genes uh, to produce the cattle without horns, right? So like this, they have produced many transgenic animals which will produce desired character. So methods of creation of transgenic animals. So transgenic animals are normally created using three techniques, three different methods. One is DNA microinjection, second one is embryonic stem cell mediated transfer, and the third one is retrovirus mediated gene transfer. In the first method, that is DNA microinjection, so what they do is they take fertilized egg and they are going to inject the gene of interest from other organism. And now what happens, of course they are going to inject the transgene. Now transgene is the gene from other organism, so they will inject it into many eggs, I mean fertilized eggs. So the gene is not going to get incorporated into the host DNA all the cases. So but few of the eggs will take up that transgene, and now we have a fertilized egg with the transgene, the foreign gene in it. So they are going to put it back into the uterus of the organism where it will develop into a full-fledged animal. So that is how the first technique is used, that is DNA microinjection, to produce the transgenic animal. Now second method of producing transgenic animals is use of embryonic stem cells. So in simple, we can understand it like this. So they get the stem cells from the blastula stage of embryo. So these are the blastocells. Now these blastocells, now they will use the recombinant DNA technology to insert the transgene into those blastocells, and it, the transgene will succeed in getting incorporated into the host cell of some of the blastocell cells. Now, since the blastocell cells are totipotent, they will develop into the new embryo. So they will make it to develop into a new embryo, and then they will transfer this embryo into the uterus of the host organism, where it will develop into a transgenic animal. So this is the second technique. The third technique is the use of retroviral vector. Okay, so this is a technique where they are going to infect. First, using recombinant DNA technology, they will insert the foreign gene into a retrovirus, right? Now, this virus, which is not virulent, because they will make it non-virulent, and they will insert the foreign DNA or foreign gene into the the retrovirus, and that retrovirus is going to infect the embryonic cells. So they will make sure that this virus is going to enter into the embryonic cells. Once it enters into the embryonic cells, since it is the retrovirus, it is going to get incorporated into the host DNA. Now, what is the host DNA? What is the host here? It is nothing but the embryo, and this embryo is transferred into the organism's uterus, where it will develop into a transgenic animal. So this is the third technique. So these are three different techniques which are used to produce transgenic animals. Now, let us understand one more concept, that is monoclonal antibodies. In short, we call them as MAB. Now, in immunology, you are going to study about the immune response. So one type of immune response is humoral mediated immune response, where the plasma cells are going to produce specific antibodies against a specific pathogen. Say for example, virus A enters into the body. So virus A infects the body. Now the immune system of the body is going to identify that A virus, and then it is going to produce anti-A antibody. I mean, the antibodies against that particular virus are going to be produced. So those antibodies which are specific for that particular virus can be considered as monoclonal antibodies, because they are all same and they are going to attack only that particular or specific antigen or the pathogen. Now, monoclonal antibodies are very helpful in fighting the infection or fighting the disease. So they are naturally made in our bodies. Now, you just think of a situation where the person's body is not producing sufficient number of antibodies, or the time taken for synthesizing or producing antibodies is long, and if ready-made antibodies can be given to the person immediately after the infection or when the disease is in its earlier stage, so the person will recover very quickly. So that is the concept of uh, monoclonal antibodies. Now monoclonal antibodies are produced using the biotechnology tools, and a large number of monoclonal antibodies against a specific pathogen or specific disease are produced, and they are used in the treatment of disease. So monoclonal antibodies are antibodies that are made by identical immune systems. I already told you they are produced by the plasma cells of the immune system, and monoclonal antibodies have monovalent affinity and bind to one particular epitope of an antigen. So monoclonal antibodies can be produced in specialized cells through a method now referred to as hybridoma technology. Now what is this hybridoma technology? Uh, Here what they do is they will say, for example, I was giving you the example of virus A. Now what happens if virus A infects the body? Our immune system is going to produce the antibody which is specific against that particular virus, right? That means if I get infected by virus A, in my body I'll be having antibodies, specific antibodies against that particular virus. And to treat the other person, right? So they want to produce more number of such specific antibodies. So what they do is they will infect a mice, right? So they are going to infect the mice with the disease causing organism. Now the mice immune system is going to produce the specific antibodies against that, right? So they have taken that specific antibody, so they will get the plasma cells. Now this is important. So who is producing the antibodies? The plasma cells. So you know that the B cells. So plasma cells are nothing but activated B cells, and they are the ones which secrete, which produce the antibodies. So they will collect the plasma cells from the mice blood, and they will hybridize it with a myeloma cell. Now what is this myeloma? It is a cancer cell. You know, cancer cell is immortal, right? So in this technique, when this hybridization is done, so they are going to get a new cell which has the property of a cancer cell. Cancer cell means which is immortal. So it will go on multiplying for many generations without causing any death. So there are no cell deaths. So it can continuously divide for many generations. So that ability is obtained from the myeloma cell. And since they have introduced plasma cells in it, it also goes on producing monoclonal antibodies. So in the laboratory, scientists can produce very large quantities of such monoclonal antibodies to treat a particular disease. So this is called the monoclonal antibodies and their application in the field of medicine. Now that's about the session where we understood the concept of biotechnology, transgenic animals, and monoclonal antibodies, right? Okay then, thank you.